हॅलो, अनम्यूट करा मॅडम. मॅडम तुम्ही अनम्यूट करा.
ओके.
सर्व
Shall we start? Ubay sir, shall we start? Ubay sir.
हा मॅडम.
Yes sir.
तुमच्या ह्याच्याप्रमाणे चालू करा काढचं.
Okay sir, thank you. So good afternoon everyone. I am Dr. Seema Deshpande, Associate Professor in Microbiology at DBF Danan College of Arts and Solapur. On behalf of Danan Institution Solapur, I welcome you all for this national webinar on molecular biology and evolution of novel coronavirus, organized by the Department of Microbiology, DBF Danan College of Arts and Science. In association, we have organized this webinar with Microbiology Society of India. About 550 participants across India, they have joined this webinar. Thank you. Thank you for joining us this afternoon. I hope you all are doing well and are taking good care of yourself and of community. Little bit housekeeping before we begin this webinar. All the participants, they are requested to mute their video and audio. To get the e-certificate, feedback form is mandatory, the link of which will be provided to you at the end of this webinar. And if you have any questions or queries, you can put these in the chat box. We will be discussing those after forwarding to the speaker at the end of this webinar during question answer session. So we begin this webinar with, as our tradition, with DAV anthem. May I request all the participants to stand in honor of DAV anthem.
अविरल निर्मल सल सदय ज्ञानदायिनी ज्योतिर्मय हो चहु दिशि उद्घोष अभय दिए विजय जय दिए विजय जय प्रबल प्रवाहमयी नित नूतन जीवन दायिनी सदा सनातन वेद प्रणीता परम पुनीता ये धारा अक्षय दिए विजय जय विजय जय दयानंद से प्रेम भक्ति ले हंसराज से त्याग शक्ति ले धर्म भक्ति का राष्ट्र शक्ति का हो दिन मान उदय दिए विजय जय दिए विजय जय प्रेम शांति इसके तट ठहरे सघन शांतिमय प्रबल क्रांतिमय लिए अटल निश्चय दिए विजय जय दिए विजय जय.
Thank you. Thank you all. Now be seated. Now moving on to, I request Professor Dr. D.G. Kadam, Head Department of Microbiology, DBF Danan College of Arts and Science, to throw the light on the objectives of this webinar.
Good afternoon to one and all. Again, I welcome all the participants. The resource person of today's webinar, Dr. Hamida B, our principal of our college, Professor Dr. B.P. Ubale, and all the organizing committee members to this webinar on molecular biology and evolution of novel coronavirus, organized by Department of Microbiology, DBF Dayanand College of Arts and Science, in association with Microbiology Society. On this occasion, I thank Dr. Hamida B from Usmania University, Hyderabad, for accepting our invitation as a resource person for this webinar. Friends, we are all facing deadly situation of the COVID-19 pandemic. COVID-19 emerged in China and it has spread globally, and it has hit badly, impacting all its activities and resulting into mental, social, and economical unrest all over the world. So far, the genus Beta coronavirus of the family Coronavirus has been responsible for 3 major outbreaks that include SARS epidemic in 2002 to 2003 in China,
Hello, Deshpande madam.
Yes.
Sir, network problem.
Okay, there is some problem with Kadam sir's side. So we will shift over to the next part of this webinar. Now, heading.
Madam,
Okay.
just once again try to call Dr. DZ Kadam so that he can complete his.
Okay, sir. Okay. काय करेल सांगू? हॅलो. हा ओके सर. मिसाळ सर. Hello, Mr. Sir.
बोला madam.
Already 100 participants are there. You remove some so that
Okay,
because that's why disconnected. Kadam sir.
Okay, madam. No, wait.
I'm extremely sorry for the technical difficulties.
Madam also announced that those participants who are failing to join, kindly join on YouTube.
The participants who are not able to come on the Zoom, they can also listen this webinar on YouTube. We have already provided you the YouTube link. Please join through
Kadam sir
YouTube.
join.
Hello, Kadam sir. Hello. Kadam sir.
Sir, check their microphone.
Hello.
Join.
Nisar sir.
हो madam.
100 participants are just remove somebody.
नाही.
Kadam
Join
sir
झाले
has joined.
तर नेटवर्क प्रॉब्लेम आहे त्यांचा तिकडे.
Kadam sir, are you listening?
Kadam sir, please continue.
Kadam sir,
Kadam sir,
make your
please
mic
continue.
unmute.
Now,
Please
no,
continue.
now I'm audible.
Yes, you are
Okay.
audible, sir.
Okay, sorry participants. This due to technical problems, this happens. So I will continue. So far, the genus Beta coronaviruses of the family Coronavirus has been responsible for 3 major outbreaks that include SARS epidemic 2002 to 2003 in China, caused by SARS coronavirus 1 with several hundred deaths. Then Middle East respiratory syndrome epidemic in 2012 by MERS virus and hundreds of deaths. And the present SARS coronavirus pandemic in 2019. During current COVID-19 pandemic, more than 15 million people are affected with more than 6 lakh deaths worldwide. In India, more than 1 million confirmed cases are recorded and about 30,000 deaths, and the spread still continues. As we all know, that the SARS coronavirus, being a novel strain of its family, it has presented new challenges before us. As on today, there is no vaccine available in the market, all being under various stages of the testing. A few companies remarkably took efforts and claimed to bring out the vaccine shortly in near future. However, it seems that the vaccine would take little longer period than they expected and longer than the claims of the companies and what is given by the media. Till then, we have to rely on preventive measures only, such as use of masks, physical distancing, and use of sanitizers. For those infected persons, the symptomatic treatment only would help. We all know that this virus has tendency to undergo mutation from time to time. We do not know and we can't predict the kind of the mutated strain of SARS coronavirus 2 that will evolve in future. Under such situation, we will have to live with COVID-19 for several months or even several years. In order to tackle the disease and characterize the virus, multidisciplinary teams are undertaking basic and applied research with objectives to decipher the origin and evolution of the virus, as well as to undertake development of diagnostics, drugs, and vaccines. In order to combat this corona pandemic war, we should be consistently be alert and should have deliberations and discussions on this disease and its causative agent. With this view, Department of Microbiology, DBF Dayanand College of Arts and Science, in association with Microbiology Society India, has organized this webinar. Hope this webinar will help to acquire some new knowledge through the presentation by today's resource person. Thank you.
Thank you. Thank you, Kadam sir. Now, moving on, I request Principal Professor V.P. Ubari, DBF Dayanand College of Arts and Science, Solapur, to briefly introduce about our college.
Good afternoon to everyone. Have a nice day. Be safe, take care, and do some social work related with the awareness of COVID-19 among the society. I feel very proud to hear with Dr. Hamid Abi, Assistant Professor in the subject Microbiology, Usmania University, Hyderabad. The DBF Dayanand College of Art and Science was established in the year 1940 and the microbiology in the year 1980-81. Department of Microbiology is doing a lot of work related with the life sciences as well as in the present scenario, the coronavirus COVID-19. In the February itself, the Department of Microbiology released the flex code, conducted the webinar on COVID, actually live or face-to-face interaction by conducting national conference on such type of pandemic diseases. Then this is the second virtual conference or webinar. This today we are having about 21 activities of such type of webinar series. Today parallelly there is one more webinar with data science for the science students of mathematics, computer science, then mathematics, and those who are having interest, the teaching faculty and researchers like that. Microbiology is working for the betterment of the society on a various platforms. I would like to very proudly quote here that the few students are working as Corona warriors or Yodha, and they are giving service to the society in the form of various uh, parameters, collecting of the swabs, then the home-to-home surveys, and the pharmacist pathological lab, and a lot of involvement is there of the microbiology students. The college this year received a overwhelming response to the UG admissions in the subject microbiology. After the establishment of microbiology department in the college in the year 1980s, after 30 years, we received unexpected response to this subject. This indicates that there is a demand of the students to know about such type of things, and life sciences is doing a lot of things for the betterment of the society. Today's webinar on molecular microbiology and evaluation of novel coronavirus COVID-19 with the resource person Dr. Hamida B is only possible with respect to the efforts executed by the head department of microbiology, Professor D.Z. Kadam, then Dr. S.N. Deshpande, Damble, Sainath Kamble, Survase, and I'm very proud to say that the first of all, our Dr. Mrs. Mushan Madam uh, said few words about Hamida B and his association and his research. And due to the collaboration and cooperative and continuous exchange of ideas and thoughts with each other and the efforts of the Mushan Madam, today we are having this webinar. I am sure that the participants, attendees, researcher, faculty, students definitely get something fruitful related with such type of diseases and how to come over. In the society, there are a lot of, we can say, unwanted things in the mind of the society, whether this is the airborne disease or it is spreads in from the air, whether there is mutation or continuously who and other medical societies are adding the n number of symptoms, and there is a confusion and stress to the society. And for that, to relieving that, I am sure that this webinar will be very highly helpful. I request all the participants to feel free to ask their questions, to put their doubts, or they ask the questions. So the expert and the subjects from the faculty from the department as well as the resource person can try to clarify the some things. And I hope that these deliberations will make fruitful to each and everyone. On behalf of the DABCMC New Delhi, Dayanand College Solapur, my faculty, students, stakeholders, alumni, I heartily welcome to Dr. Hamida B, and I request her to remain associated with our microbiology department. So in future we can go to exchange of ideas, thought, knowledge, and we can provide much more to the students as the college is running a MSc microbiology, as it is a research center under the guidance of Dr. D.Z. Kadam. Few students awarded the PhD degree. Also, research projects are there, add-on courses are there, skill development courses are there, and lot of things are there. And we would like to have the association of Hamida B Madam with the college, with the microbiology department. Once again, welcome to the madam and best wishes to the organizer, the staff, faculty, and students. Thank you to one, and have a nice day and have a nice session. Thank you to all.
Thank you, Bahir sir, for your nice introduction. Now, virtually, we are felicitating our chief speaker, Dr. Hamida B. Madam.
Thank you.
Welcome, madam. Once again, welcome. Dear friends, we know that the whole world is facing COVID-19 pandemic. This infectious disease outbreak is due to coronavirus of the family coronavirus. The world has never seen this virus. It appears that the virus transferred to human from animal host by acquiring new mutation. And therein lies the problem. We now have the enemy that we can't see and that we don't know much about it. How do we fight such an enemy then? The only way is to study and learn more about it. So the scientists all over the world within a short period of time made significant discoveries that have led us towards a better understanding of this novel coronavirus. And these findings could help to translate into developing novel therapeutics or preventing strategies. Let us know more about the molecular biology and evolution of novel coronavirus from today's speaker, Dr. Hamida B. I have great privilege and honor to introduce to you a very learned, enthusiastic, multifaceted woman who has devoted herself for the research field, Dr. Hamida B. So Dr. Hamidabi is working as assistant professor in Department of Microbiology, Usmania University since 2007. Before joining Usmania University, she traveled to Canada to work as postdoc research scientist in the Department of Plant Science, University of Manitoba, Canada. Then she joined Mumtaz Degree College, Hyderabad as lecturer. Her research focus on beneficial bacteria for plant growth promotion and disease control. Besides this, she is also interested in characterization of microbial surfactants with application to agriculture, human health, and pharma. Up till now, she has published 26 research papers and has written 5 book chapters. She has also presented her research articles in about 60 different conferences. Besides this, she is recipient of many awards and fellowships, notably Research Appreciation Award, Distinguished Scientist Award, Young Scientist Award, and Postdoc Fellowship. So, we are very fortunate to have such an academically sound, research dedicated speaker with us. I am hopeful that her talk will definitely be useful and informative to us. Once again, I welcome Dr. Hamidabi Madam and request her to reveal the molecular level story of novel coronavirus. Over to Hamida Madam.
Thank you. Am I audible?
Yes, Madam.
Yeah. Good afternoon, everyone. Respected Principal Sir, Professor Vijay Kumar, and Head of the Department, Dr. D. Kadam Sir, and Deshpande Ma'am, and all other delegates, the teachers and the lecturers, and dear students. And I also want to share that, Dr. Lakshmi Mushan is my school colleague, and we are very good friends, and we come from the same place. So we have been associated for a long time, like from childhood to degree, we were together, and then lot of doing different courses and all those things. So always we discuss subject as well as family things, and we are getting it's long association of her. And about the present talk, that is the COVID-19. Yeah, it's already there. I need to share again.
No, ma'am. Share again.
Okay. So, actually, yeah. And I'm a microbiologist, but and I teach virology for the PG students from the past nearly more than a decade. But I was a little bit hesitant to speak on COVID because a lot of webinars were coming up, and everyone was asking, "Will you speak?" Because I don't have practical, hand-on experience or working with the viruses. though I teach, that is theory is different and practical. That's what most of our students always say, that ma'am, we want more practical exposure than theory, because theory can be read, it may be understood, our information is already available. Sometimes it goes inside, sometimes it doesn't. But when you have practical experience and practical exposure, that is where the students, or even as faculty or teachers, we are also more, conscious to talk when we have practical information. So mostly I present about microbes and the surfactants and where I am good at. But again, I was also thinking, because I teach virology, I should also know a little bit more about this coronavirus. And that was the chance I took up. Okay, when Lakshmi Mushan said, why don't you speak? I said, okay, let me take it as a thing. And I had to learn a lot before coming here and sitting before you in the webinar. That is the first part. And this thing is, I would not touch each and every aspect. As we know, most of, it's like more than 6 months, this coronavirus has come, and we are listening a lot. And every month or every day, today we listen new things about the virus. Either we think of the people, getting infected. And so I'm not giving the already available information on the Google, the symptoms. So I try to be, focus it a little bit on where the story of this coronavirus might have started, and did the researchers miss it? That is, when I say researchers, maybe the virologists or the people who are working, thought, okay, everything is going smooth, because we are at a global village and day-to-day activities are very fast. So, somewhere we missed it. And now when the pandemic is there, now we are thinking that as biologists, microbiologists, or geneticists and ecologists, or epidemiologists and clinicians, we have to do a lot of the things. Okay. So now I switch on to the presentation, and I'll be speaking about this novel coronavirus. And I think all of us know, especially for the students, we call it as COVID-19 because it's a coronavirus infectious disease, and it's in the year 2019 it's been reported. And what you can see is like, these days, many of the, even a kid can, even if you show a small picture, even a kid of 3 to 4 years say it is a coronavirus. So I want also to show the electron micrographic image, how it really appears. It's been taken from the Wikipedia. And most of the information I have taken is from either, Wikipedia, different YouTubes, and the different research presentations. So I acknowledge all the support, taken from the different research articles. Wherever possible, I try to acknowledge them. So coming to the coronavirus, my presentation will be about the origin of the virus and its evolutionary strategy. And I'll give information about the structure and its genome organization. And I didn't make it very hi-fi, and also because I wanted the students should get some insights about this virus. So I tried to maintain balance. So I think all of you will be coping up with me. And at the end, if you're able to understand a bit about the origin of the virus and its structure, and what are the different vaccines available about these things, so that will be, I'll be happy that, okay, students could catch few points from my webinar. And as I said, we are all microbiologists, and always we speak about microorganisms. So I'll add a tinge, like these days, what we are doing to a cup of milk, we are adding a tinge of turmeric, and we are making decoction or kada. We are trying to take tinge of every spices available in the kitchen. The purpose is we are trying to see that we get, protected from this coronavirus infection. So I'll also add on few slides that as microbiologists, in addition to wearing of mask and giving different suggestions, the microbiome associated with us, it also plays a significant role in the COVID-19 fight and maintaining the immune system. Yeah. So coming back again to the students, most of the students, again, from the students' thought or mind, I'm saying, is this important? Do we need to sit for an hour to listen to this webinar? We are listening so much on TV, and so much information is already available. Colleagues are saying, neighbors are saying, and friends are saying, why again we should sit for 1 hour? Is it only to get the e-certificate for the purpose? Or this information is going to be available to us, useful to us. So how can the information useful in our world? So that thought should come in the student's mind. Yeah. So either we are studying the structure of the water molecule or the mitosis or many more aspects. Most of the students, they do, am I audible?
Yes, yes, yes,
Yeah,
madam.
okay. So most of the students, like either they go for the, I mean, taking of the different subjects, either arts or science, anything, mostly with the GPA thought, okay, will I get merit, will I get through these examinations, will I get the degree? But always I would say that if this, whatever you're trying to read today or understand, most of the information may go, I mean, may not be clear, but you can jot down those points, and with time you may be, as biologists or as researchers, you may be working on those things. So the topic may be not be, I mean, sometimes it may be a new topic, but you should get motivated. You will get motivated only when you think this present, whatever I am going to learn today will be going to be useful in tomorrow's world. So purpose is the research, the seeds of research should be sown in the younger minds. So we should, because a lot of, see, that is also the reason we do not have good virology laboratory facilities to start working with the virus on day one. So we were searching in and around in, national wise, which lab is available, where we can talk and where we can really do the, because we need the BSL-3 facility to work on these viruses. So we have to see that, we get motivated towards the research. And biologists, it's not, don't think only the microbiologists, all the biologists and microbiologists can do a lot. So when I am trying to teach something, as I said, you try also to learn something from the presentation. So coming back to the talk about the origin of the viruses. So if you look into the thing, how does a really a pathogen? Because we know, if you look, you're already looking onto the slide, there's the name of the Zika virus, there's the name of the MERS, there's the name of the Ebola, dengue, chikungunya, and I think most of us, have heard about these viruses. Even the students, specifically, I'm saying, the moment you might have come to a higher class, you might have heard about these viruses. So viruses, how do they infect or how do they start? Either they need the vector, and most of the viruses, they might be associated with different animals. So they may be associated with the wild animals, they may be associated with the domestic animals, and from wild animals, the spillover can happen to the domestic animals, and from domestic animals, it can also, because we are associated with, with the domestic animals like cats, dogs, and different things. So they can also spill over to the humans, and they can infect. And this is how we have different viral diseases and the different pathogens, emerging worldwide. And as of now, we are worried about coronavirus, but there are in the past, if you look, people have suffered from dengue, chikungunya, Ebola, and also Zika virus. Okay, so is it fine?
Yes, madam. Go ahead, madam.
Yes. So what type of animal to animal, animal to human transmission? Though the slide is showing the shape, one second. The shape of the monkey here, I don't mean it's only the monkey. I just wanted to I have taken it from the animal to human viral transitions, an evolutionary successful one. And it's a recent article from the Journal of Molecular Evolution. So it could be any animal. So we have to see, as I said, we have millions to billion contacts of the humans and the animals. Okay. And it is not that every animal will cause us an infection, because specifically whenever a virus has to infect us, we need the receptors. So the most common reason is the absence of an appropriate receptor, because the way the virus goes and sits or attaches. So we need a sort of tropism for the virus to attack the cell. So absence of the appropriate receptors, the virus will be destroyed. So when we come to the second, the animals, they may occasionally infect the humans, but as long as it is not causing any, sort of infection and they are not characterized, most of the infections are not unknown to us also as of now. And if you look into the third, that is the rabies virus, I think all of us know. The virus, that infects one person, and it's lethal also, but human to human transmission is not possible with the rabies virus. And coming to the next part, if you see here, viral transitions are characterized by mild infections. And there is also transmission. When it is having mild infection also, we are not so worried. There is low probability of human to human transmission. That is the example of the cowpox virus. And coming to the next part, if you look here, so we are more worried when the infections are lethal and when there is fatality, when there is mortality. So, if you look into the MERS virus, there is a Middle East research. I'll a little bit come back to those viruses again. It is a Middle East respiratory syndrome virus. And the fatality percentage, also varied based on the strain. And coming to the, if I just rounded up this F and H because this is like the type of common cold virus. So I mean, there are many type of common coronaviruses which also cause the common cold. But as long as the infections are mild, we are not worried. So it may spread to many people, but their like infection is very mild. But coming to the next, I mean, if you look into this H part, this is, I mean, the SARS, that is a severe acute respiratory syndrome coronavirus 2. It has high probability of human to human transmission, and also it is leading to fatality. So coming to the Ebola virus, it's also high probability of transmission and also high fatality rate. So this is how they are much, I mean, virus and animal, I mean, that is animal to human viral transmissions were happening from a very long time. And we are trying to understand about those different viral infections. So if you look at these things, as I said, this is nothing but the Wuhan, I mean, the seafood market in China where people roast different types of wild animals and all those things. I mean, they consume it also in the part of the, not only for the diet, but also for the sort of traditional medicine. So that is the reason, I mean, this spillover of the virus from the wild animals to the other, I mean, intermediate host and to the humans have happened. And that is how, I mean, in addition to, we know that, I mean, we are loss of biodiversity, that is, we are, I mean, going for deforestation and all those things. So that could be one reason, climate change, and many other aspects also have added up to this transfer of the viruses from the animals to the humans. So if you look at these human coronaviruses, though we say that, okay, now we are saying coronavirus, okay, from 6 months, we are every day in and day out, we are talking about this, but it's not that they just came back 6 months or 10 years, 1 decade back. Molecular clock analysis indicates that these viruses diverged from, I mean, the closely related coronavirus hundreds of years ago. So they were there. That's the only thing is we are listening because, as I said, whenever it comes to, because as humans have always used this sentence, that is, we exploit all the existing thing, and at the same time we want like to protect the human population. So always we'll be doing different types of research and all those things for the humans. So we are also speaking a little more about this coronavirus because of the COVID-19 disease. So mutation and as well as adaptation, because this is how the coronavirus has evolved along with the host. So I just want to say one thing here. Suppose, see, if you think of a, I mean, like a nail, or I mean, when you touch a hard surface or a nail pricks your leg, you, I mean, have a sort of response because we are, I mean, live. So that is how the viruses and the host, that is, we are, it's not some, you know, when you take some inorganic or some metallic substance, it's not going to react. So that is how the viruses, and also we are also trying to evolve. And this mutation and adaptation had driven the co-evolution of the coronaviruses and their host. So till now, if you look back into the literature, there, I mean, now this is the 7th, that is SARS-CoV-2, what we are trying to say is the 7th one. And already 6 have been existing for human coronavirus 229E, human coronavirus HK, this for the Hong Kong, where the, I mean, reports have been come up. Human coronavirus OC43, this 229E and 43 are present global wise. And human coronavirus New Zealand, I mean, from the New Zealand 63. And we also heard about the, I mean, the head department saying about SARS, that is a severe acute respiratory syndrome, SARS-CoV, and also the Middle East respiratory syndrome. So if you look at these things, go back to the evolution, these have been existed around 500-820 years ago. And this you can see OC43 around 120 years ago. So like they have been, as I said, if you look back into the molecular clock analysis, they have been present hundreds of years ago. But I mean, as we are worried about the coronavirus 2 and the infection, we started looking back into the literature and giving information about all these different coronaviruses. And if you go back to SARS-CoV-1, it's like around 8,000 plus cases have been reported and 774 deaths. So the story has begun, but again it ended. So again, people are not so curious or anxious to make about the work on the vaccines. And that is how we have ended with the SARS-CoV. And MERS, Middle East also, respiratory syndrome, where still, I mean, infections are occurring. But you can see here, I mean, the cases are very less. So these two still resulted in the pandemic. And more information for the students, you can refer this article. And if you go for the classification, we have 4 different, like the alpha coronavirus, beta coronavirus, gamma coronavirus, and the delta coronavirus. I mean, and that infects the different birds and also the mammalian host, that is including the human and also different animals. So this is how, I mean, bat is the reservoir of the most of the, I mean, coronaviruses. So if you look here, I have again given the lineage of the human coronaviruses. So where did they originate? I said most of them are bats, and sometimes we also have intermediate host. It is some, I mean, maybe camelids. And again, we have NL63, where again it was the bats. And again, we heard about the HKU1 and OC43, where they said the possible origin could be the rodents. So this is how we have the lineage of the alpha coronaviruses and the beta coronavirus. And beta coronavirus, this is about the MERS, and also we have the beta coronavirus SARS-CoV. So as I said, most of these coronaviruses likely all emerged from bats and were transferred to humans. Okay, this is not an article of 2020. Make sure in mind, people were reporting much of the research on coronaviruses in 2012, 2013, 2015, till 2019. But we were not, as I said, until and unless it comes for us as a sort of, infecting or causing some, damage to the humans, either in any aspect, either it is the climate or the disease or anything, we are not much worried. So these all articles I just picked up to show that, these all coronaviruses, or more than 500 coronaviruses have been identified in the bats in China. And people say, that is the researchers, or you can say the people, ecologists, people who are doing research and giving data, they say there are more than 5,000, because we also know as microbiologists, metagenomic studies also have added up much information about the different bacteria or microbial diversity, either it is the bacteria and the viruses. And they say that about more than 5,000 bat coronavirus diversity is existing. So as I said, the bat is the primary host, that is, it has the coronaviruses. And as I said, wild animals too, some other animals, it is the domestic animals, either it is because of the seafood market, or either you have seen already, we know about the Wuhan, or it could be any other source from the wild animals. It spill over, the viruses gets infected, carried or transmits into the domestic animals, and from domestic animals, it's ultimately we humans get this virus transmitted. So if you look into that is the sequence homology and the genome, that is because a lot of research, as the head of the department and other people are also saying, because this 6 months, if you see, most of the articles are available on Google, and you can read also. Most of the articles are available for all the researchers to read and gather information. And if you look, that both the SARS coronavirus, that causes the, in 2003, and the present SARS coronavirus, it shares about 80% nucleotide identity. And if you look, that is, if again I have just taken the SARS-CoV-2 and the SARS-CoV, where in the SARS-CoV, that is, we said, it could be the dogs or the civet cat that could be as the intermediate host. Whereas now we, people, you see, it's still, not clear. It is still, a lot of papers or articles are coming out. But people say that because when they try to make the phylogeneticity of the SARS, they're taking the different genomes, what they have seen is 96.2% nucleotide homology of the SARS-CoV-2 with the bat coronavirus RATG13. Okay, and similarly, they also have seen that, it also had the similarity of about, 85 to 92% with, that is pangolins. These are nothing but the scaly animals where the Chinese use for the traditional medicine. So that is the one reason they are saying it could be, it's not still clear that it is an intermediate host. People, if you look back into the literature, they say that it might have infected the bat, and from the bat, it might have gone to the pangolins, and pangolins from, again, it might have gone back to the bat, and that is how it could have transferred to the humans. Okay, so they are different, they are not still, no clear conclusion has there that it is from bat and from two pangolins to humans. So it could be from bat to humans also. So that could, that sort of information is available in the literature. And if you look where the viruses, attached to the human cells, that is I'll come back to this receptor binding domain shortly. So they have done the sequence of the receptor binding domain of the pangolin coronavirus and also the bat coronavirus. Here in the receptor binding domain, they have seen that the higher amino acid identity is with the pangolin coronavirus when compared to the bat coronavirus RATG13. So this is how conclusions are there for the evolution of the virus, or you can say the transmit of the virus from the bats to an intermediate host, which is still yet to be defined, and then to the humans. So till here I was saying about how the different coronaviruses were present, and people have made the study, and now we have a lot of information has been gathered about the coronavirus too. And if you look into the, why do we need to understand the structure? So most of the students might be thinking that we know much about the virus structure and all those things. That is heavy about the helical symmetry and the icosahedral symmetry and all those things. So why because based on the structure, the special features, there are specific features we can also target for the vaccine development. So coming back to the virus morphology, as I said, coronaviruses, they are they form envelope and they are like pleomorphic to you can say about spherical particles, and is the size of the virus 100 to 160 nanometers in diameter. And the genome, that is the viruses either have DNA or RNA as a genetic material. So they contain a positive sense single stranded RNA genome of size around 26 to 32 kb in size. And if you look into these things about the genome, the 5-terminal end, it encodes for the polyproteins, which forms the viral replicase transcriptase complex. And then we have the different, I'll go step by step into the structure of the virus. We have the membrane protein and we have the spike protein. You can also see this is the main reason, spike protein, which allows the virus to attach to the human cells. That is a receptor. At the same time, this crown-like structure also has given the name coronavirus to the virus. There is a crown shape. And we have the envelope protein and also the ribonucleoprotein. So you can see this. This is because of the crown-like structure, the name corona has been given to this virus. And we have 4 different structural proteins. That's the spike protein, envelope protein, membrane protein, and the nucleocapsid. So coming to the first part, that's the spike protein. It has you can see here, I think all of you, this is a spike protein, and this is little bit in clear. It has 2 functional subunits, S1 and S2. And this spike protein attached to the, it needs a sort of receptor, that is the ACE, angiotensin converting enzyme. And you can see this angiotensin converting enzyme is present in the lungs, heart, kidneys, and intestine of the humans. So that is the reason we are also listening many people who have been infected and gone with the severe stage, they are passing away with the kidney failure. So because we have these ACE2 receptors in the different parts in the humans. So if to understand about the spike protein, it has the receptor binding domain, and it recognizes the angiotensin converting enzyme to receptor and helps to attach to the host cell. So the spike protein binds to the ACE2 receptor with the same affinity, and you can say in the, as I said, the SARS-CoV, it is 20 times more, with more affinity, it is binding. That is also the reason it is spreading so easily from human to human. So coming back to the 2 parts, that is the host cell receptor. So S1 binds to the host cell receptor, that is ACE2, and S2 helps in mediating the fusion of the virus and the cellular membranes. Because it is how the virus to enter into the host cell and create an infection. First, it needs to go and attach, because it's binding to the host cell or and make its entry. It's also used as a target for different making of the different vaccines and also the medical treatment of the COVID-19. And again, as I said, is it a new virus? Just came back 6 months? No, and only, now we see about the ACE2 receptor. No, if you look back into the Nature article and the Journal of Virology of 2012, people are saying that, characterization of a bad SARS-like coronavirus that is using ACE2 receptor. So, they were existing, but as I said, not much information, because we were not having any cases or any reports about the different infections caused by this bad coronavirus. We were not studying in detail. So you can also see one more article in there, 2012. People are saying that angiotensin converting enzyme is being used as a receptor for the coronavirus, and that is from the bats. So I spoke about the S protein. Coming back to the next, membrane protein. It is abundant on the virus because the viral needs a surface and a sort of shape. So that membrane protein helps, and it is also allows the assembling, because when the virus infects the cell, it has to make its entry, it has to multiply, it has to assemble, and then it lyses the host and leaves. I'm not speaking about the lytic and the lysogenic cycle. Thinking that most of the students know it. So I'm just going into the next step about the virus membrane protein. And coming back to the E protein, it starts with the envelope protein, and it's the smallest of all the structural protein and plays several roles. It also helps in assembling and releasing of the virus particles from the host cells. During replication, what you can see is it is largely localized at the site of intracellular trafficking, mostly because see, this is an RNA virus. So the replication happens in the cytoplasm. So intracellular trafficking also happens specifically at the endoplasmic reticulum and the Golgi apparatus. So if you look back to these two specific proteins are very important. They play, as I said, spike protein is very important for the attachment of to the host surface. Similarly, membrane and E protein are very, envelope protein play a critical role in turning the, because we know the viruses, when they infect the host cell, they use the host cell apparatus to make its own replication, that is its genetic material, and again go back and infect the neighboring cell. So what does these mRNA proteins do? They turn the host cell apparatus into workshops where the virus and our own cells work to make the new viral progeny. And underneath the surface protein, we have the envelope. So we said it's envelope virus, and this envelope has, it's like a waxy barrier containing fatty molecules. So now I think most of you might be thinking that always every 10 minutes, or they're asking us to wash the hands whenever we go out and come in, or whenever we are touching any surface or anything that has come through courier or through vegetables, or somebody is getting us some vendor is getting us and giving some material. Why? Because viral envelope, it has the fatty molecules, and washing with the soap, that is the first preliminary step where you can eliminate the virus without entering or without you getting contact with the virus. So this is how the viral envelope protects the virus outside the host, and also it allows the, anchors the structures needed for the virus to infect a cell. And coming to the genetic material, you can see it's the single-stranded RNA, positive sense genome. And I think in the Baltimore classification, it comes into the fourth group. I'm not going much into the details, as I said. I'm not teaching the virology class. I'm giving the information about the viral genome. And this underneath the capsid, we have the nucleocapsid. And when this nucleocapsid, it binds to the RNA and also helps in the replication. And one more thing about the N protein is, that is the nucleoprotein, is it's multifunctional. In addition to helping in the replication, it inhibits that is a host cell defense mechanism is also inhibited by the nucleoprotein. So this is how about the different structure of the virus and how it enters into the host cell. So you can again see the spike protein, and this is how just I put the host cell, and you can see the cell membrane, and that is how the virus enters using this angiotensin converting enzyme as a host cell receptor. And you can also see here, this is a virus, and this is the how you have the angiotensin converting enzyme receptor and how it comes and attaches. And this is about the S1 and S2. And sometimes the virus can have a sort of direct entry. That is how our host cell helps it, because we have certain proteases, and that enzyme will be released, and it clears the spike protein and allows the membrane fusion and injects the viral genome to enter the host cell. In other conditions, endocytosis, that is how most of the bacteria enter the host cell. Similar to that, the virus, it is being surrounded by the cell membrane and then buds off into the host cell and enters the host cell and causes the infection. And to understand the size, we said that this 25 kb or 30 kb virus, which is not seen, is causing us so much problem. At the same time, if you look into the depth of the genome organization, it itself has so many features that it is able to multiply rapidly and also cause the infection. So we have, if you can, I'll just repeat the virus structure. So we have the spike protein, then we have the membrane protein envelope. This is our nucleic acid, that is the RNA, and then we have the nucleocapsid. So if you look, this is about the 29,674 base pairs of the genome, which has made us worried today. And if you can look into that, we have two things. That is, we have non-structural proteins and the structural proteins. So we have ORF 1A, and we also have ORF 1B. And if you look, 2/3 of the genome consists of the open reading frames that are translated into this peptide-like protease and also the 3CL protease. And then we have the non-structural proteins. And if you look into these things, we have this NSP12, because the genetic material is RNA, and this RNA-dependent RNA polymerase. This is more important for the making of the viral replication and using of the viral replication strategy. And the rest, when I said about the structural proteins, that's what I was just explaining about the different structure of the virus. That is the membrane protein, envelope protein, nucleoprotein, and the spike protein. So how the virus enters, as we know, the genetic material is RNA, and the replication happens inside the cytoplasm. So you can see the replication strategy. Now I have told you about, how does the virus look like, and what are the receptors on the host cell. So, and how it enters, either a direct entry or through endocytosis, and that is how it dumps its genome. Okay, there's the positive sense RNA. Because of the positive sense, the ribosomes can directly read it, and it can make the next strand and make subgenomic RNAs And that subgenomic RNAs can be responsible for making of the different non-structural proteins and the structural proteins. And those things happen in the via the rough endoplasmic reticulum and the Golgi apparatus. And after making those things and assembling of the different viruses, through exocytosis and to release, it comes outside the host cell and again infects the next cell. So this is how the viral replication happens inside the, you can see, Golgi and the endoplasmic reticulum. So if you can see here again, though I said it's only 25 to 30 kb, but you can see it's making so many different proteins, that are helping it to attach, that are helping it to, multiply, that are helping it to assemble, and that are helping it to infect the different cells and the different hosts. And coming to the next word, okay, about the vaccine development. And as we are listening so much in the media, it's very hype, thinking that August 15th, we got the independence, and we are also going to get independence from coronavirus. I'm not contradicting or making any statement. It's not so easy, because as researchers, all of us know, to get a vaccine, it's a very long procedure. And if I say that for the undergrad student who has come into, we say so much about Louis Pasteur, and different, not only Louis Pasteur, about the different people who have started about the vaccination and how many deaths initially happened because of smallpox and all those things. Though we, because as I said, these days, because for us, food is available over the, just we make a phone call, like a Zomato, we get the food. Just sitting online, we recharge our phone. Because we got habituated for the things to happening within minutes to hours. We are also thinking vaccine is going to come within hours to days, but it's not an easy task. Most of the researchers know, most of the teachers who are sitting also very well know that it's not an easy task. But still, because it's causing high fatality, spreading human transmission is rapid and people are suffering. So people, different researchers, that is, have come together and are trying a lot to get the vaccine in a short period of time. And that is how nearly around more than 160, different companies, or you can say different research groups, are involved in making the vaccines. And it's I have taken it also from a literature where it says 24 candidates are in clinical evaluation and 142 candidate vaccines are in preclinical trials. So I'll just try to explain a little bit about the vaccines. So I think most of us have taken, when there is an infant or child, in the first week we take the BCG vaccine, and then we say, okay, you have to go for the DTP. That is how different vaccine, the days will be told by the doctor and the infant gets. So similar way, so at that time, all these vaccines are also developed with different things. So any vaccine development, it needs a sort of preclinical testing, and the sort of preclinical testing in the sense it needs, different cell, animal cell lines, and it will take long time, because that's also the reason I said for the SARS and MERS, had the infection spread for more time and to more people would have focused on the vaccine. But because it came and not infected many people, and also went away along with the wind, so people did not focus much. But now they are going back to the literature and collecting information of SARS-CoV and trying to do fast work on the coronavirus 2 vaccine. And coming to the clinical testing, you need phase 1, phase 2, and phase 3 trial. Again, it will take long time. In regular, it may take 3 years, 5 years, as I said. But now for the coronavirus, people are simultaneously doing different experiments and different things intact, and they are trying to make it short to less than an year. I would not say even 6 months, 9 months, it's less than an year. So what can be the virus candidates? As I said, people will be targeting the spike protein in the virus. And the virus, it can be the viral vector based, it can be vaccine, protein based. Many different candidates can be worked on in order to control the viral infection. So we have viral RNA vaccines, DNA vaccines, recombinant vectors, and we have inactivated vaccines. And as I said, though we are saying less than a year, you can see here, because we have to practice all. I said we do not have many laboratory facilities within our country to do this type of laboratory research work. So we need good practices at the lab to work. Then we need the phase 1, phase 2, phase 3 trial. We also need the FDA approval, because it can't be directly dumped, saying though the fatality is more or infection is more, we can't say, no, you just after the 3 months trial, we can we can give it to the humans. No, it is not possible. And at the same time, we also need the large scale production of the virus. And then we have to also make a study about the immune system and the immunity. So this is what I already said. We can use either recombinant vectors or the mRNA vaccine. That's what the Moderna claims it as mRNA vaccine vector. And we also use the attenuated virus. So ultimately, purpose is whether our body, that is, can because of these vaccines, can create the immunity and neutralize the viruses producing the antibodies. That is also the reason, if it's not recognized and it's not neutralized, the viral infection will be there, and that again it causes the infection. So this is about the weakened viruses, how we can weaken the virus by passing from one generation to other, induce mutation, and then allow it to weaken the, that is alter the genetic code and weaken the virus. So inactivate virus is nothing but we can use certain chemicals or heat and make the virus into inactivated form and try to make the vaccine. So there are many strategies available, people are working. And as I said, 42 candidate vaccines are there. This is what Serum Institute of Pune using, from the AstraZeneca company, is trying to work and expect, because the phase 3 trial is already done. They are claiming that they will be the ones who will reach the market shortly, but we have to hope for the better things. And I was also speaking about the Moderna virus, where the people have taken as a, nanoparticle encapsulated mRNA, which is also, claiming to be done with the till the phase 3. But a lot of studies are yet to be done. And if you look back into the Indian companies, we have, I'm not reading about all the other companies. So Cadila Healthcare is also there. Bharat Biotech has also become very much hyped these days. It's one of the company located at the Genome Valley, Shamirpet in Hyderabad, where the whole virion inactivated thing is there. But you can see still it is in the phase 1 and phase 2 trial. And these are all the other companies or the other people who are in the line, or you can say making the race towards making of the vaccine. And just adding a bit about the, because the Moderna vaccine, which is so much, because if you just put on the TV and you can see the lady taking up the injection and they say about the Moderna vaccine. So it's about the mRNA 1273. It's nothing but a lipid nanoparticle that's being encapsulated, and it is based on the SARS-CoV-2 spike, because that is where the virus goes and attaches. So it is in the pre-fusion conformation stage, and that is how the vaccine is being made, and it's also in the phase 3 trial. And coming to the Bharat Biotech, you can also see it's like only phase 1 and phase 2 are done, and that also 13 July was the date given in the web, the information available, and the sample size was only 1,000. That is only, around 1,000 plus people were given this vaccine. So, and how anyhow, because ICMR and National Institute of Virology Pune are also involved along with these things, we can hope, because we should have a positive hope that something is coming to rescue us at a short time. But we also should understand, we have to have the clarity, because before the vaccine reaches the market. So till here, I was trying to gather information. I tried to gather information about the coronavirus, and that's the causes the COVID-19 disease and how it was present. There is a version of the human coronaviruses was there from long, but we were not knowing much, because only from once the you can say WHO declared it as an initially as an epidemic and then a pandemic, people are worried, and then now we already know 6 months down the period how many people got infected in our own country, in our own state, and are suffering. So as microbiologists, can we do something to protect ourselves in addition, because we said we are trying to give information to the people about maintaining social distancing, washing of the hands with soap, so and wearing of mask. Can we also do give some more information and we are saying you have to take this spices and this tea and that tea and little bit of vitamin C, little bit of vitamin D. So I'll also add a tinge that how our microbiome also may play a role in the COVID-19 fight. I'm not going again about the metagenomic study and what is microbiome, expecting that little bit about the microbiome, that is as microorganisms, and we are having different microorganisms associated with us, that we call it as a microbiota. And this information has been come out because of the initially when they done the human genome project and the sequence, they found that more than the human genome, we also have the genome sequence from the microorganisms. So that is how it turned into human genome project, has been turned into human microbiome project, and that is how we have a lot of hidden microorganisms which are present. Not only, it's not don't think as microbiologists we have only microorganisms on the surface, on the head or on the hand, but wherever you are sitting. So that is how the metagenomic study has given a lot of information about the different microorganisms associated with us. So this is I just took a picture for you to understand that how many different microorganisms. It's like a network. We are different microorganisms are associated with us, and they're also responsible for us to maintain good immune system, immune homeostasis, and good health. And it also says that more than the cells present in our body, we may have microorganisms, or few some literature also says we have microorganisms and the cells also more or less similar in our body. So this was one more thing where I could catch the review article from Dhar and Mohanty. And you can see here gut microbiota and COVID-19, possible link and implications. So they said there may be a link between the gut microbiome and health. See, as I said, this is a new thing and most of it is at-end research. I mean, we can provide information, make different hypotheses, and until and unless done on different experimental stage and we come to a conclusion, we cannot make an authentic statement. So most of the times you'll be listening to your gut, right? I mean, at least now specifically, when the moment you're sneezing or coughing, first is you're worried. You sit for a while, then you say, no, it is already in the monsoon time. Because of the rain, it might be common cold. That's what you get worried. Is it a common cold? That's a seasonal flu or the seasonal cold what we get, or it could be because of the coronavirus. So as I already said, the coronavirus attacks angiotensin converting enzyme, and I said it's present in the lungs, it's present in the gastrointestinal tract, it's present in the heart, and also the kidneys. And people show symptoms of not only the cold, cough, but also diarrhea, nausea, and vomiting. And different studies have been done. People said that people had gastrointestinal presentations only, and more percentage of people had both gut and respiratory illness. So at this, I am speaking about the people who are, had infection and who had more viral load and who had infected. So, and it also says that it took longer time for the people with the digestive symptoms to clear the virus. So what could be the role of the gut microbiota in modulating the immune response in COVID-19? There could be different reasons. It says that gut microbiota, they regulate the innate and also the adaptive immune system. So the naturally present microorganisms in our gut or the intestine, there's the intestinal commensals, the normally present bacteria or microbes, they secrete certain peptides, and that peptides will be competing for the nutrients and the habitat, thereby maintaining the, that is, they will allow the naturally present commensals to sit there and allow the foreign coming path, that is the coronavirus, to remove. So this is how the gut microbiota and immune homeostasis may have a back and forth relationship and help in the fighting of the infectious diseases. In addition to that, gut microbiota signals are also known to tune the immune cells to make certain pro-inflammatory things and also the anti-inflammatory response. So this is how you can see that the good gut microbiota and the good and this good microbiota also comes from the different food. So these days we are saying we should not use outside food. We should take good food. We should also take food which is having different probiotics. We should take different fruits. So that is how you are trying at the same time to maintain the good microbiome in order to avoid the, give a tough time to the coronavirus infection. So it's also called as gut microbiota and gut, because, and one more thing I want to say is even our lungs also have different microorganisms. Initially people thought lungs do not have any microorganisms, they are sterile, but it's not that. Even lungs also have microorganisms, and it says that from the gut and the lungs also, there is a sort of exchange, like how we are exchanging, sitting over one, I'm sitting at my place, you're sitting, but we are trying to exchange our ideas or information about coronavirus. Similarly, the microorganisms sitting inside the gut give certain signals to the lung and vice versa. So because of that also, it helps in the combating this coronavirus infection. And we are saying that most of the people are the elderly people and the immune compromised people and people with different comorbidities like diabetes and also hypertension. So how this happens, because these people will be already taking drugs, and so this type of infections and all those things are also because of the microbial imbalance already that has been happened. This microbial imbalance, it's also called as the dysbiosis. So there is an information that intestinal microbiota exerts on the lungs, and that's called as the gut-lung axis. And the gut-lung axis, it is bidirectional. That is, there is some inflammation happening in the lung, then it also affects the microbiome that is sitting in your gut. So similarly, because the microorganisms there get changed, so again you may have inflammation. That is also the reason we are saying more about the cytokine storm is also one of the reasons we are having this problem. So unbalanced microbiome will also lead to different inflammation, and that is how coronavirus is waiting. How do I get a chance to attack the host? Not only going and sitting and attaching, not only lungs, wherever I have got the receptors, I'll go and attack. So if we are giving a tough time by maintaining the good microorganisms and good and healthy microbiota in our gut, which comes again from the different food and the diet we are taking, we may give, a sort of, like we can say, labs for the coronavirus to infect us. So that is how I mean, we also, like it says, because of the, as I said, combined inflammation, that is the cytokine storm. This is also the reason we are having the great damage than the virus. Okay, the virus is coming and attacking, but beyond that, we have the, I mean, our own immune system responding, and that is how we are also having the problem with this coronavirus. And coming back to some more information about the different bacteria and COVID-19 severity, there is a Caprobacillus species, there are different Clostridium species. So they say that Coprobacillus species is magnifying the ASC2. I mean, it's a study in the mice, angiotensin converting enzyme 2 expression in the mice. So that is how, as I said, the different type of the microorganisms sitting in your gut will allow or magnify or help the virus to, infect. Similarly, there is also the about the good bacteria, about the Alistip species and the Fischlibacterium. They are said that they are helping in participating in the tryptophan serotonin metabolism. That is essential for maintaining the gut immune homeostasis. So that is how taking good food and taking care of your microbiome, I mean, good fiber food. So I just should, I mean, these days you might be thinking, I have not taken the burger, I have not taken the, finger chips, it's wrong. But I would suggest that at this time, in addition to using the mask, in addition to maintaining social distance, you also take good fiber food, good protein food, good probiotic food that may indirectly also help you. I'm not saying this is only the solution. This may also help you in maintaining the, healthy microbiome and also overcome the COVID-19 disease or overcome, a little bit, what you can do is coronavirus infection may be inhibited. So this is how it is also from the same article I have taken. In addition to, I'm not saying, probiotics, prebiotics are going to add. I'm saying in addition to whatever you're trying to give, that's the different drugs and the different treatment modalities, try to also take good food, that is a homemade food. As microbiologist, and I know because the Microbiology Society also conducts different competitions, and students have worked a lot on the probiotics, prebiotics, Rangoli competitions, and all those things. A lot of information is all. Mostly, I would say that Maharashtra students are more interested in microbiology, and even we try to motivate. But I see, because where Professor Deshpande is president, a lot of activities are there for the microbiology students. So as Madam Mary Curie said, be less curious about people and more curious about ideas. As microbiologist, I always tell to my students that, whenever, irrespective of the class, I take care of your health, take care of your microbiome, because if your microbiome is happy, surely your immune system is happy. And if your immune system is happy, you are going to overcome many infections and the diseases. So I also like this. When I was going through the national webinar, that is the login details, I just found that I will wear mask and will save you. You wear mask and save me. So I was just thinking, then I was asking ma'am, and it's a caption given by the principal sir. So I liked it, and I just wanted to share a small video. So just for 2 minutes, I'm not taking much time. I don't know whether I have students, they were, one second. One second. You're able to see the screen?
Yeah, madam.
Yeah. Hello. Yeah, now I'm done with the presentation, and I think hopefully I can give information wherever I have, learned a little bit about the coronavirus. Any questions?
Thank you, madam.
Yeah.
Thank you for your nice information. And you have explained everything in a very lucid language, because most of our students, they are listening this webinar, and that would be I think more useful or beneficial for the students also. Not only for the students, but some of the informations were there in your speech which were new to us. Especially I liked your microbiome, how the microbiome it is responsible to develop the immunity, and therefore whatever the vaccines they are coming up in the market, whether that vaccine will work on individual or not will be decided on the basis of the microbiome which is present in that individual. So you have thrown the light on the how the evolution of the novel coronavirus has been there, and also you have thrown the light on the molecular structure, as well as whatever the vaccines they are being developed, discovered. You have thrown I think light on many of the aspects, and therefore this lecture was very very useful, beneficial to all the participants, I think. And many of the participants, they are commenting that your demonstration is candidly very nice. So even there are many questions coming up in the mind of the participants, so they are waiting to ask these questions. So I hand over now the question answer session to Dr. Shripad Survase.
Yeah.
Thank you, Dr. Deshpande madam. Am I audible?
Yeah.
Yes. Good afternoon, everyone. I am Dr. Shripad Survase, and I am first thankful to Dr. Hamid Abi for such a knowledgeable lecture. Now it's time to a question answer session. I will forward some of the questions from participant to the guest speaker. Let us start with the first question, that is from Pawan Pandit on Zoom meeting. Okay. Why do coronavirus have such a high mutation rate in such a less time? Less than 2 decades, it has changed more than 2 hosts.
Yeah.
Dr. Hamida,
yeah. See, viruses, if you look back into the evolutionary strategy, it's a convergent evolution strategy, they says. And that is how, the viruses, these coronaviruses have been, mutating. That is, it's not for the first time. Like if you look back into the literature, and now this mutation, because we have genetic reassortment. If you look back into the influenza virus, which is very well known and most of the students learn, genetic reassortment happens in the, most of the viruses. So it's a, natural phenomenon. What you can also think is, yeah. Okay. What you can think is maybe because of the, different, and viruses, they multiply at a very fantastic rate. Like you can say the billions of copies will be produced in the small time. So that is also one reason why we, it's not that coronavirus is having mutation. The viruses, they mutate rapidly.
Okay. Let us ask the next question. That is from Saili. How to prevent attachment of spike protein to the ACE2 in any chemical way after entering in a host? Is it possible? How to prevent attachment of spike protein to the ACE2?
I don't think, because chemical way it's not. I have not seen, at least I have. As I said, I was browsing from less than a week about these things, but I have not seen any chemical method. Because most of the people will be saying, like, if we can get some chemical where we can gargle, and like how we are saying gargling with but the common cold also we gargle with the salt and all those, turmeric and all those things. So similar, that is also the suggestion given. And only some chemical like betadine, some is there, but I don't think there is a known or a recommended chemical available for the virus not to attach to the ACE2 receptor.
Okay, the next question is from Pranav Belsre. How bats are reservoir for the virus?
See, like as I said, now microorganisms are associated with humans, with animals, with plants. So they are the reservoir of the different coronaviruses. Literature says most of the coronaviruses, they originate from the bats.
Okay, thank you, madam.
It could be
The
you can say sort of
Delivered
So doesn't cause the infection to the bats
information.
or any
Delivered of
effective
news.
infection to the vaccine. Okay. The next question is from Vijay Lakshmi Vija, that she told that, ma'am, as you told that vaccine production takes 3+ years normally, but now corona vaccine production giving news that it has a on trial on humans. Then is it 100% effective, or is it for a temporary prevention of the virus?
See, as two things are there here to explain. Viruses are, they mutate. So getting the correct candidate vaccine is a, very important phenomenon. And the second thing is, will we get 100% thing and all? Because now it is still a study in the phase 1 and phase 2. We still do not know. Because if you go back and just Google a little bit about the already candidate vaccines, just one or two, not many. Even if you just look back, as I said about the, Oxford vaccine and all those things, there are a lot of just I'll give a small example. Just if you have to go to a X-ray, they'll very specifically say that is if you are pregnant, you should not, get exposed to the X-ray. So like there are a lot of rules and regulations first to take up the volunteers for the vaccine. And then the vaccines are, phase 1 and phase 2 will be given, and the detailed study has to be done. As of now, sitting together here, I can only say what candidate vaccines can be taken as, what researchers are working and how much time it may take, but will it give a 100% thing? Because see, in this time, will we get herd immunity? We do not know. Because even if you look back into the 1918 Spanish flu, it took 2 years, 1918 to 1920. In this duration, I'm not giving you a negative, or very goosebumps saying that all of us will get infected, but who knows how many of us will get infected, and herd immunity itself will answer, and then vaccine will come. So ultimately, if you look back, plus 1 decade, like now we are in 2020, inshallah, if you think of 2030, at that time, people may think, just speak, okay, like how today we speak about, I'll give an example, like Mycobacterium tuberculosis causing TB or typhoid. If somebody eats some non-veg food outside or something which is uncooked and they say they got typhoid, oh, okay, doctor will say you got it diagnosed after 1 week because the fever is not gone. And then you, okay, it's typhoid. So do all these measures, take this type of food, take this, drugs or antibiotics, and then you'll come. So down the decade, it may also have the similar thing, because vaccine is one thing, but people are also working effectively on antiviral drugs, which I did not again still touch. So which will come, see, ultimately is if something protects the, that is increase, the people, whoever are infected comes out and gets relief immediately. Either it's an antiviral drug or the vaccine, or with time it's the herd immunity. It's all, question to debate, or I cannot say concluding answer for this.
Okay. The next question is from Zeba Parveen. Is COVID-19 virus can transmitted through fly insects if they
No.
sits on the infected places, or like this? Or
No, as of now, we do not have any vector type of any, because if anything is, different people might be doing different experiments, but I did not see any information on vector. Even food also, people have some questions. What I have seen is through food also, is it getting transmitted? But no, as of now, we do not have any information that it is being transmitted in that manner.
Okay. The next questions we are asking from YouTube Live. It is from S. Adib. That is, if virus is undergoing molecular variation,
yeah,
then what will work? A vaccine or a multiple drug therapy?
Yeah, and just now I answered saying that I can't make a concluding statement what is going to work as of now. I said, that is the reason I also added the microbiome thing in our thing. Yeah, because I said I did not touch on the antiviral. See, one thing is I did not touch on the symptoms, transmission status, clinical aspects, because much is known from this past 6 months. So I thought, let me again little be focused, because students should, because if we repeat the same thing, they may not really show interest in listening for because I know the receiving end of the students is very less, and listening for like 30 to 40 minutes is like half for the students, maybe not for the faculty.
Okay. The next question is from Siva. Is there any changes in genome of virus place to place? from location
Place to
location
place. I have. One of I said actually I have taken few slides from one of our zoology. Pardon? Yeah. One of our zoology professor who is Dr. Radhakrishna. He has worked little bit on the. They said there is little bit variation. Not all the viruses in India have been sequenced. In Hyderabad and Karnataka they have done, and it's reported in database. There is slight variation. I have not read much on that, but some information is already there on the strains from Hyderabad and strains from Karnataka.
Okay.
Maybe if you check on the NCBI, because Gandhi Medical College and Usmania University, this group of Dr. Radhakrishna, they have done a little bit on the sequencing on the viral genomes from collected from few patients.
Okay, thank you, madam. The next question is from Devdatta Das. Will the virus lose its virulent power in the near future with a more mutation?
That's what I said. That's time of. I said if you go back and look into history, either you take the example of smallpox, or if you take the example of Spanish flu, which itself took around 2 years. So we do not know. Like I said, herd immunity can, because people either through taking of the vaccine or people getting infected and immune, and few people who are strong having strong immune system, they also can help in this thing. But it's like today we cannot tell, because people from Spanish has tried, people in Iran, they say it's a herd immunity. But if we look back to our own country, we have still the peak of that is a graph being raising. So we have not yet reached any of that. As I said, no, these days we want everything over the desk. Like again, our kids, we made them habituated like that. So yes, that's the reason.
So, there is a last question from YouTube, that is from Devdatta Das. Is herd immunity possible if the virus keeps mutating to new strains or with cases of reinfection?
So I'll again take this into two things. So herd immunity being possible if it gets mutated and it loses its virulence, that itself is enough for us. But if it's still being virulent, it may take a little bit of more time.
Thank you, madam. Solving the questions of the participants. The questions are ended. Now one can ask a question online directly from a Zoom meeting. I will hand over the mic to the Dr. Deshpande, madam.
Those who want to ask the question from Zoom can raise their hand.
Yeah, can I ask? May I ask?
Yeah, madam.
Yeah, I just wanted to know, we were aware of gut-brain axis. Could you please throw some light on gut-lung axis as to how would it reflect?
Yeah, even as I was saying, I also speak a little bit on the microbiome in the classes. So initially I was actually, making slides and information only on the gut, and brain axis and how it gives signals. But when I was looking back to the coronavirus and the article by Dhar and Mohanty, so how they said the possible link. So they have shown that, because even our lungs are not sterile and they are also having the microbiome. So it says like there is, if some inflammation is there in the lungs, so that signals will be passed on, and that is how even our gut microbiome may get disturbed. So dysbiosis may happen, and they say it may be bidirectional also. So it's well explained in a little bit in this the article. It's a journal of. I have given the details in the presentation. So if somebody is interested, I can pass on the presentation. Also, you can look back into the last slide, so where more detailed information is there about the gut-lung axis.
Yeah, madam, thank you. It was a very coherent, poignant, and a very lucid presentation. Although we being teachers, you kept on emphasizing, students would like it, but I think we teachers also, some aspects were really novel to us. Thank you, especially microbiome.
Yeah, thank you, ma'am.
Actually,
I can speak a lot on microbiome, but I thought, okay, when Lakshmi Mushan was saying, I said, okay, let me again, because I always enjoy teaching microbiome to the students.
Absolutely, very fascinating.
Madam, I thought again invite you, madam, for giving the lecture on microbiome. Next lecture will be on microbiome by you. Okay, for our students and all the participants.
Sometimes me and Dr. Mushan tried that I would come, but because of the family and other things, like it was not, it's like before this pandemic and all, twice we thought we will make it, but it did not work out. So anyhow, we had a webinar. So these days, many things are possible because of coronavirus and COVID.
Yeah, madam. One more question can I ask? is there any study regarding those who have taken SARS vaccine are immune to COVID-19? Is there any study regarding
No, I have
those
not
who have
checked.
taken the SARS vaccine are immune to COVID-19?
No, I have, because even SARS vaccine also they give certain flu shots, and I mean they give, I mean, even in the Western countries, but like it was also not a complete study, ma'am. I mean, even if we look back onto the vaccines available, like because as I said, it started and it stopped. So people, I mean, there are certain vaccines available, but I do not have the, I mean, clear information whether this is inhibiting the. Because I think if that would have been, then flu shots have been given to all the people till now. But I have to check. I can't make a statement without checking.
Thank you, madam. Thank you once again.
Thank
Now we are coming to the conclusion part of this webinar, and I invite Kamble sir for vote of thanks.
Thank you, madam. Am I audible to all?
Yes. Yes, you are.
Respected dignitaries, guest speakers, eminent faculties and students, as we come close to National Webinar on Molecular Biology and Evolution of Novel Coronavirus COVID-19, it is indeed a proud privilege for me to propose a hearty vote of thanks to all. At the outset, on behalf of Organizing Committee, Department of Microbiology, DBF Dayan College of Arts and Science, Solapur, and on my own behalf, I would like to thank Padma Shri Honorable Dr. Poonam Suriji, President DAV College Trust and Management Society, New Delhi, for giving us opportunity to host of this event. We would like to thank Sri Maheshji Chopra, Local Secretary, Dayanand Institution, Solapur, and Secretary DAV College Management Committee for giving inspiration and support in the organization of this event. We are deeply grateful to the respected Principal, Professor VP Ubale, DBF Dayan College of Arts and Science, Solapur, for presiding over this function and enlightening address. We would like to express our deep felt thanks to our chief guest speaker, Dr. Hamida Abi, Assistant Professor, Department of Microbiology, Osmania University, Hyderabad, for delivering a very useful session and covered very important information from origin of coronavirus and development of vaccine. In spite of her busy schedule, she could spare time for this occasion. I extend my sincere thanks to Dr. A.M. Deshmukh, President of Microbiology Society India, for the great contribution in the organization of this event. I also express my sincere thanks to Mr. Deepak Misal, technical support during conducting of this webinar. We are thankful to all participants who have joined us at the event from distinct universities, institutions, laboratories from various states of India and abroad. Lastly, my thanks are due to my all colleagues of DBF Dayan College of Arts and Science, Solapur, who have worked with great dedication to make this program a success. Thank you.
साइनस कांबळे सर.
Ah, yes, sir.
अरे मुशन मॅडम चे आभार मान.
Oh, sorry, sir.
Special. Because of
Extremely
Mushin
sorry,
madam only we could arrange this webinar, sir.
Yes.
So
Sorry, sir. We are very thankful to Dr. L.C. Mushan ma'am for great contribution in the organization and arranging a guest for this webinar. Thank you, madam.
Thank you, Kamble sir. Now we are ending this webinar with national anthem.
जन गण मन अधिनायक जय हे भारत भाग्य विधाता पंजाब सिंध गुजरात मराठा द्राविड उत्कल बंगा विंध्य हिमाचल यमुना गंगा उच्चल जलधि तरंगा तव शुभ नामे जागे तव शुभ आशीष मांगे गाहे तव जय गाथा जन गण मंगलदायक जय हे भारत भाग्य विधाता जय हे
काय सर?
क्लिक करतो लाईव्ह फाईलला. जर मला फील्डला क्लिक करता क्लिक करता येत नाही.
मॅडम, thanks a lot. Very good coordination and nice organization. I am especially thankful to Dr. Hamida B. Madam for sparing his valuable time. I personally request Madam to remain associated with the college and the department, give the idea regarding the changing trends in life sciences, so that we can incorporate all these in our curriculum, as well as we can provide that to the students on add-on basis. And after, as and when situation becomes normal of this COVID-19 pandemic, you do come to the visit to the college at Solapur and interact personally with the students, have a round in the college campus to the life sciences departments, and give suggestions for the betterment of our students as well as the departments. Amidabi, you have given a very nice and fruitful lecture. As I am not belonging to the life science section, I would not comment much more on your things, but your language is too simple, lucid, confident, fluent, pronunciation, and all the content, as well as your reference quoting. All these things are nice, and I feel proud to listen you. So I remain in the meeting for throughout the